Glucose. Glucose is a simpler sugar that plays a central role in metabolism, which is the process by which our body converts what we eat and drink into energy. It is a type of carbohydrate that gets absorbed into the bloodstream and used by cells for energy. After the intake of food, the concentration of glucose in our blood rises and attains the hyperglycemic state. When it goes too high, the pancreas releases insulin into the bloodstream through beta cells of islets or Langerhans. In the pancreas, glucose primarily binds to the GLUT2 transporter, where GLUT2 transporter is a type of glucose transporter that plays a crucial role in glucose homeostasis. Insulin allows glucose to be taken up and used by the insulin-dependent tissues such as muscle cells. Glucagon and insulin work together automatically as a negative feedback system to keep the blood glucose levels stable. The insulin signaling promotes the translocation of GLUT4 glucose transporters to the cell membrane. They involve GLUT2 and GLUT4 transporters. This condition leads to glycolysis and glycogenesis.
Glycolysis takes place at all the cells. It mainly occurs during high glucose concentration and low ATP concentration and during fed state. Glycolysis is most widely distributed catabolic pathway which proceeds through fructose 1,6-bisphosphate. It is also known as FBP pathway. Glucose comes in metabolically active form, which is phosphorylated on carbon 6 by hexokinase and converted into glucose 6-phosphate. Thus, glucose 6-phosphate is the starting point of all three lytic mechanisms. Hexokinase is allosterically inhibited by ATP, citrate and glucagon, whereas it is allosterically activated by AMP, ADP and fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate or phosphofructokinase 2 is regulated by fructose 6-phosphate and phosphofructokinase 2, whereas it is allosterically inhibited by citrate and ATP. Pyruvate kinase is allosterically inhibited by ATP, alanine and glucagon, and it is allosterically activated by fructose 1,6-bisphosphate and AMP. This full glycolysis yields 2 ATP, 2 NADH molecules per glucose molecule. The final product, pyruvate, can be reduced to lactate in anaerobic condition or enter the citric acid cycle in aerobic conditions.
Glycogenesis is the process by which glucose is converted into glycogen for storage, primarily in the liver and muscle cells. This process occurs at fed state when both glucose and ATP is high and it is stimulated by insulin. Glycogenesis is essential for maintaining blood glucose levels and ensuring a steady energy supply during fasting. The process of glycogenesis begins with the phosphorylation of glucose, converting it into glucose 6-phosphate through the action of hexokinase or glucokinase. This glucose 6-phosphate is then isomerized to form glucose 1-phosphate catalyzed by the enzyme phosphoglucomutase. The next step involves activating glucose 1-phosphate by binding it to uridine triphosphate UTP, resulting in the formation of UDP glucose, driven by the enzyme UDP glucopyrophosphorylase. Glycogenin acts as a primer for glycogen synthesis, while the glucose units from UDP glucose are incorporated into a growing glycogen chain. This incorporation is facilitated by glycogen synthase, the key regulatory enzyme in glycogenesis. The branching enzyme also plays a crucial role in creating the branched structure of glycogen, ensuring that it is stored in a form that can be quickly accessed when the body requires energy.
When the blood glucose levels are low, it attains the hypoglycemic state where the pancreas responds through its islets of Langerhans, specifically the alpha cells. These cells produce glucagon, a hormone that is crucial for raising blood glucose levels. Glucagon is released into the bloodstream and travels to the liver where it binds to a specific receptor on the liver cells. The binding activates processes that with increased blood glucose levels, including the breakdown of glycogen into glucose, that is called the glycogenolysis, and the production of a new glucose from non-carbohydrate sources called the gluconeogenesis. This coordinated response helps to restore the blood glucose to normal levels. Here, GLUT2 helps the glucose to exit the liver and enter the bloodstream.
Gluconeogenesis is the process involved in the synthesis of glucose in the body with the requirement of ATP with a low glucose level. This process is initiated by the hormone glucagon. Glucagon is an hormone which is secreted by the alpha cells of pancreas. While the glucagon present in the body, the level of insulin is reduced. The oxaloacetic acid present in the mitochondria turns back to the malate by the reversible reaction using malate dehydrogenase enzyme. This is due to the oxaloacetic acid cannot exit from the mitochondria, and due to that it is turned back into the malate molecule, and it is get into the cytoplasm by the malate aspirate shuttle, and it is turned back to the oxaloacetic acid by the same enzyme which is used to convert malate to oxaloacetic acid, and oxaloacetic acid by the enzyme phosphoenolpyruvate carboxygenase it is converted to phosphoenol pyruvate molecule, and by the continuous reversible reaction it get into the fructose 1,6-bisphosphate molecule, and by the phosphofructokinase 2 enzyme it is converted to fructose 6-bisphosphate molecule, and by the phosphorylation process the phosphate molecule from the fructose 2,6-bisphosphate is removed and it is converted to fructose 6-phosphate. Then it is transferred to endoplasmic reticulum, and by the T1 transfer protein and by the T2 transfer protein the phosphate is removed and it is converted back to the glucose molecule, and it is distributed to where the glucose molecule is required in the body.
Glycogenolysis. It is the metabolic process by which glycogen, a stored form of glucose, is broken down into glucose. This process primarily occurs in the liver and muscles, and it is essential for maintaining blood sugar levels, especially during fasting or intense physical activity. The process of glycogenolysis. First, glycogen phosphorylase activation. The process begins with the activation of glycogen phosphorylase, the key enzyme in glycogenolysis. This enzyme is activated by hormones like glucagon and epinephrine, which are released in response to low blood sugar levels. Glycogen breakdown. Glycogen phosphorylase catalyzes the cleavage of alpha-1,4 glycosidic bonds in the glycogen molecule, releasing glucose 1-phosphate units. Next, debranching enzyme. Glycogen molecule have a branched structure with alpha-1,6 branch points. The debranching enzyme is required to remove these branch points. Next comes the conversion of glucose 1-phosphate to glucose 6-phosphate. This is converted with the help of an enzyme called phosphoglucomutase, where glucose 1-phosphate is converted into glucose 6-phosphate. Next is the glucose release. In liver, glucose 6-phosphate is further converted to glucose by an enzyme called glucose 6-phosphatase and released into bloodstream, raising blood sugar level. In muscles, glucose 6-phosphate is primarily used for energy production through glycolysis.
In conclusion, the homeostasis of blood glucose is a vital process that keeps our body sugar level stable, ensuring our body functions properly. This balance is achieved through the coordinated action of insulin and glucagon, which regulate glucose uptake, storage, and release. By maintaining this equilibrium, our body can provide a steady supply of energy, prevent damage from extreme glucose levels, and support overall health and well-being.